Hello everyone, I'm Sumeer and I'm working with Professor scAEGAN. Today I'm going to talk about our project Unification of the single-cell genomics data. Particularly, I'll be talking about our development method that is scAEGAN, which is the unification of the single-cell genomics data by using adversarial learning of the latent spaces. Technologies for the production of the single-cell omics data are rapidly developing, which includes various library preparation protocols and data coming from multiple modalities or multiple omics, and which leads to the complex nested batch effects in the data. Thus, a joint analysis of the single-cell omics datasets is required, and which requires a reliable data integration. The integration and the combination of the data is still an outstanding challenge. It includes different challenges such as combining different experiments, same modality, same library protocol, or the data coming from different experiments having the same modality but a different library protocols. Or we can have data coming from two different paired data modalities or two different unpaired data modalities. So here we show that a coupling of two neural network architectures is sufficient to solve a broad suite of data integration challenges, as these challenges have been previously tackled with individual methods developed for each particular challenge. So in this architecture, we use two neural network architectures, which are the autoencoders and the scAEGAN. So first, an autoencoder learns a low dimensional embedding of each experiment independently, and which respects each sample's uniqueness, protocol, and the data type. Thus, it does not force them into a shared latent space. Then next, we use the scAEGAN, which learns a non-linear mapping between these different AE representations. So we evaluated our method using the simulated data and the real data set, which includes different data modalities and the different library preparation protocols. In order to evaluate the performance of the scAEGAN on the various integration tasks, we evaluated for the first experimental setup, that is the cross-in platform and data, and with having the same modality. The visual comparisons with the translated versions of the other domain reveals a good agreement, where we have this A to B and B to A are the translated domains, and these are the original domains. So the scAEGAN preserves the transferred cell identity agreement with the original identity. Next, we tried to evaluate its performance on the same platform data having the same modality, where we have the data from the old and the young mice. And the results over here shows that the scAEGAN still preserves the cell structure in the translated domains. In the third setup, where we tried the efficacy of the scAEGAN, is the paired data having the different modalities, where we have two modalities. One is the AE RNA-seq data and another one is the AE ATAC-seq data. And the results over here shows that the scAEGAN still preserves the cell structure in the translated domains. Next, we try to quantify the integration quality, where we measure the transferred cell identity agreement between the translated domain and the original domain. And here we use the two evaluation metrics. One is the adjusted index and another is the Jaggard index. The results over here shows that the scAEGAN outperforms other methods considerably well in the various integration tasks. Then we tried to evaluate the robustness of the scAEGAN, where we removed a certain percentage of cells from each data sets and tried to see how does it affect the performance of the scAEGAN and other methods, where we use different ratio of the cells. And the results over here shows that the scAEGAN outperforms other methods as well here in this robustness task. We conclude that we develop a unifying architecture, which is an end-to-end supervised single-cell data integration method, where we demonstrate that a unifying architecture, we can achieve good performance on various integration tasks. And it can be used for various tasks on different replicates, treatments, tissues, or sequencing platforms. Thank you for listening, and I would like to thank all our collaborators who have been part of this. Thank you.